Hello everyone, I am Dr. Krishna Yadav, and this video, Part 3 of Unit 1 for MPharm in Pharmaceutics, covers the Computer-Aided Drug Delivery System, or CADDS.
We begin with an overview of statistical parameters in pharmaceutical research, which guide us in summarizing, interpreting, and validating data. This visual shows the process from data collection to analysis. Statistical parameters enable the shift from mere observation to scientific evidence. Visually, two identical-looking products require statistical validation of formulation, consistency, and biological variability to prove they are identical. Statistical parameters are numerical measures for central tendency, variability, precision, accuracy, and model performance. They are vital throughout drug development, from formulation to PK modeling, ensuring quality and data-driven decisions. These critical questions underpin pharmaceutical research. What is the average? How much variation exists? Is the model accurate? Is the result reliable? Answering these drives formulation, assay validation, dissolution studies, stability testing, bioequivalence, and pharmacokinetic modeling. Statistical parameters address key scientific questions across 6 areas: central tendency, average, dispersion, variation, relationship, correlation, model adequacy, shape of distribution, and inferential statistics, reliability. Statistical parameters are quantitative descriptors from experimental or clinical data that characterize distribution, dispersion, relationship, and reliability of observations. Without them, decisions rely solely on observation, not scientific evidence, impacting regulatory submissions and acceptance criteria. Statistical parameters in pharmaceutical research are classified into 6 categories. These are central tendency, dispersion, shape of distribution, relationship, model adequacy, and inferential statistics. They guide the process from collecting data to making decisions. Chapter 2 focuses on measures of central tendency. These measures describe the, quote, average or typical central value of data, forming the foundation of all pharmaceutical statistical analysis. The 3 main measures are mean, median, and mode. Mean is the common average. Median is the middle value, less affected by outliers, and mode is the most frequent value. Mean is widely used in assay and dissolution analysis. The mean, or arithmetic mean, is the sum of all observations divided by the number of observations. It's the most common measure of central tendency, representing the central location of data. It is vital in assay and dissolution studies to check batch quality against label claims. The median is the middle value in an ordered dataset and is robust against extreme outliers, making it a truer central estimate for skewed biological data. Mode is the most frequent value, useful in categorical data, but rarely used in quantitative analysis. This chart summarizes central tendency measures. The mean is the most common average, while the median is robust against outliers. The mode is the most frequent value, typically used in categorical data. Chapter 3 covers measures of dispersion. These describe the spread of data around the mean. Variability is critical in pharmaceuticals, as high variability can indicate poor quality or instability in a product. Range, the simplest measure of dispersion, is the difference between the highest and lowest values in a dataset. For tablet hardness values of 5, 6, and 7 kilograms per square centimeter, the range is 2. The limitation is that it only uses 2 data points and ignores the distribution pattern of intermediate values. Variance measures the average squared deviation from the mean, while standard deviation, or SD, is the square root of the variance. Low SD indicates high precision and good uniformity, essential for reproducibility, method validation, and batch consistency. This is an example of calculating standard deviation. Given tablet content of 98, 100, and 102, the mean is 100. After calculating the squared deviations from the mean, the variance is 2.67, and the standard deviation is approximately 1.63. Coefficient of variation, or CV%, expresses standard deviation as a percentage of the mean. It's crucial for comparing datasets of different magnitudes. A CV% below 5% is generally considered acceptable precision, while a CV% above 10% indicates poor precision. This chart summarizes the measures of dispersion. Range is the spread between the highest and lowest values. Variance is the average squared deviation, and standard deviation is its square root. CV% is the ratio of SD to the mean, useful for comparing variability across different datasets. Chapter 4 covers measures of distribution shape. These describe the symmetry and peakedness of the data, which is critical for choosing the correct statistical test for analysis. The main measures of distribution shape are skewness, which addresses asymmetry, and kurtosis, which measures peakedness. Understanding these helps in selecting appropriate statistical methods. Skewness measures asymmetry. Positive skew has a long right tail, and negative skew has a long left tail. Skewed data often requires log transformation before analysis, especially in pharmacokinetic data. Kurtosis measures peakedness, important in assessing biological variability. High kurtosis means a sharp peak, and low kurtosis means a flat distribution. Chapter 5 focuses on measures of relationship. These are essential for studying the connection between two or more variables, which is vital for formulation optimization and understanding drug behavior. The measures of relationship are correlation coefficient and regression coefficients. They quantify the strength and nature of the linear association between variables. The correlation coefficient R measures the strength and direction of the linear relationship between two variables. Values range from 1, a perfect positive correlation, to 0, no correlation, to -1, a perfect negative correlation. Regression coefficients, or slopes, represent how much the response changes when the independent variable changes by 1 unit, used heavily in formulation optimization. This diagram visually represents different types of correlation coefficients, ranging from perfect negative to perfect positive, illustrating how tightly data points cluster around the regression line. Regression analysis helps predict the value of a dependent variable based on an independent variable. It is crucial for understanding how formulation variables impact product quality. The difference between correlation and regression is key. Correlation measures the strength of the linear relationship, while regression defines the nature of the relationship, allowing for prediction. Chapter 6 introduces measures of model adequacy. These measures evaluate the performance and accuracy of statistical models, ensuring that predictions are reliable and meaningful in pharmaceutical research. Model adequacy measures include the coefficient of determination, or R-squared, and the standard error of regression. R-squared measures the proportion of variance explained by the model, while the standard error estimates the average distance between observed and predicted values. The coefficient of determination, R-squared, shows how well a model explains variability in the data. Its range is from 0 to 1. A higher R-squared value indicates a better model fit, with values close to 1 showing excellent explanatory power. This chart illustrates how R-squared values reflect the data spread around the regression line. A value of 1 indicates a perfect fit, while a value of 0 indicates no linear relationship.
To better understand our data models, this table breaks down the key differences between 3 essential statistical tools: correlation, regression, and the coefficient of determination. While the correlation coefficient tells us how closely two variables move together, this chart shows how regression measures the size of that effect, and R-squared measures the overall accuracy of the model.
Adjusted R-squared corrects R-squared for the number of predictors, helping prevent overfitting in multiple regression. RMSE, or root mean square error, measures prediction accuracy, where a smaller RMSE indicates a more accurate model. This visual summarizes the measures of model adequacy. R-squared measures overall model fit, while adjusted R-squared accounts for multiple predictors. RMSE measures the average prediction error, ensuring the model's predictive power is reliable. The standard error of regression estimates the average distance between the observed data points and the regression line. This is crucial for understanding the uncertainty of predictions. The Akaike Information Criterion, or AIC, balances model fit and complexity. A lower AIC indicates a better balance, making it widely used in pharmacokinetic modeling to compare competing models. Chapter 7 focuses on inferential statistical parameters. These are used to draw conclusions about a population from a sample, acting as the bridge between experimental data and real-world decisions. Inferential statistics parameters include the standard error, confidence interval, and p-value. These measures quantify the reliability of results, enabling evidence-based decision-making in pharmaceutical development. Standard error, or SE, indicates the precision of the sample mean. It is the standard deviation divided by the square root of the sample size. A smaller SE means a more reliable mean. This chart visually represents how the standard error decreases as the sample size increases, illustrating that larger samples lead to more precise estimates of the population mean. The confidence interval, or CI, is the range in which the true parameter lies with a certain probability. For example, a 95% CI from 98 to 102 means we are 95% confident the true mean is within that range. It's used in bioequivalence testing. This diagram illustrates how repeated sampling generates confidence intervals, visually demonstrating the confidence level. The narrowness of the interval relates to the precision of the estimate. The p-value is the probability that an observed difference occurred by chance. If p is less than 0.05, the difference is statistically significant. If p is greater than 0.05, there is no significant difference. It is essential in comparing formulations.
To determine if a new drug formulation is genuinely better, or if the test results are just random chance, researchers rely on the p-value. This visual flow shows how hypothesis testing leads to a final statistical decision. This infographic breaks down the concept of statistical significance, showing step-by-step how a calculated p-value determines whether the difference between two pharmaceutical formulations is actually meaningful. Thanks for watching. Please share and subscribe.